The brain is a complex organ, controlling all functions of the body, but also interpreting processes in response to external information. Activity in the brain is a result of action potentials, electrical signals through nerves, generating, sending, and receiving information. These action potentials are able to be measured by electrical systems within implanted or external electrodes. Brain-machine interfaces aim to record and process these neural signals for use in human robotics. Currently, BMI methodology involves invasive surgery to penetrate the brain tissue for electrode implantation. This is done by a craniotomy, which is the cutting of the skull to access the brain tissue below. This method involves high signal strength and sensitivity, however, is highly traumatic for the patient. Non-invasive methods involve the placement of surface electrodes, though this results in high levels of noise and low signal strength.
A novel mechanism to measure these signals is utilizing the vasculature of the brain and stent technology to address the challenges associated with the direct implantation of or surface contact with electrodes. A stentrode is a mesh tube with an electrode array. Through a minimally invasive surgical procedure, the stentrode is inserted through the jugular vein into the brain via catheter angiography. A team at the University of Melbourne developed this technology and successfully showed proof of concept. The clinical study demonstrated safety and effectiveness of the stentrode with high-density microelectrodes distributed on the self-expandable structure made of nitinol.
Nitinol forms a natural oxide layer, improving the biocompatibility and corrosion resistance, as well as shape memory characteristics. However, the current concerns with using nitinol in stentrodes is the long-term resistance to fracture and durability. Considering these challenges, our design was developed with the following specifications in mind. Firstly, durability. Secondly, lead breakage. Thirdly, the minimization of signal noise by controlling charge density and impedance. And lastly, safety, including sterilization and reducing tissue damage with surgical invasiveness.
Our design maintains the closed cell type structure, but builds on the early stentrode concept by incorporating more electrodes into the system. This design reduces wall shear stress, improving hemodynamic factors and prevents vessel rupture, while the placement of the electrodes at the most complex struts allows the material to maintain its flexibility. The material choice of nitinol is due to its conductive, biocompatible, fatigue-resistant, and shape memory properties. Current problems with the use of nitinol and stentrode technology lie in the occurrence of fractures. Our team aims to change this by increasing the fracture resistance of the material via electrode polishing. Benefits of electrode polishing nitinol include improving the corrosion resistance and increased durability, as well as performing functions such as deburring, precise size control, and ultra cleaning.
The target of the stentrode is the superior sagittal sinus as it runs adjacent to the motor cortex. The surgical procedure to implant this device is minimally invasive and has little risk compared to craniotomies. Mounted over balloon and implanted via a catheter, it enters the body via a leg or groin artery and threaded to the correct location. Once arrived, the balloon is inflated to expand the stent. Once positioned, the balloon is removed. Over a period of approximately 8 days, the stent becomes incorporated into the vein, increasing stability, safety, and proximity to the target tissue. Compliance to regulatory standards in terms of safety, sterility, and manufacturing is of the utmost importance, and the design is able to do this whilst also showing efficacy. As the stentrode device poses significant risk, our team will require an investigational device submission approval before being able to gather data to support a PMA submission. In order to ensure that the safety of the device is proven, animal and clinical studies will be conducted in accordance with good laboratory practice as outlined in 21 CFR 58 and have electrical safety parameters determined by ISO 14708. Safety, manufacturability, and biocompatibility of nitinol has been proven by past literature which indicates that other neurological medical devices comply to FDA regulatory standards. While currently there are no stentrodes on the market, clinical trials are being performed to assess the safety and efficacy of other similar devices.
The major regulatory challenge for our device is the approval of the novel manufacturing process of the nitinol used, as the device has a similar mechanical design to devices that are currently in clinical trialing phases of regulatory approval. With this in mind, our team will undertake testing to ensure that the electropolishing process has compliance to safety, efficacy, quality management, and manufacturing standards such as ISO 13485, ISO 11135, and ISO 10993. Verifying reliability over device lifetime and structural integrity is something our team is working towards.